is Megan Mahoney, and I'm the Director of Scientific Affairs and Training Programs at BiocanRx. And I'm very pleased to welcome you to our 8th iteration of the Summit Speaker Series, which was developed as approximately a bi-weekly series to bring our community together to have talks by our funded network investigators and our HQP and our patient community to tell you a little bit more about the funded research by BiocanRx. So I would like to welcome our 2 speakers for today's session and thank all of the participants for attending. There are quite a few repeat attendees, and so that's always great to see. So for today, I'll be introducing the talk first with its title, and that is Increasing Gene Therapy Vector Production Using Viral Sensitizer Molecules. And we don't have the next session lined up just yet. The details are still being worked out, but I would like to highlight that the next session will be promoted through our social media channels and also online. And so we will be posting information about our website in the chat box. So without further ado, I'd like to introduce our 2 speakers for today's session. So the first is Elena Godbout, and she is a senior research associate at the Ottawa Hospital Research Institute, working with J.S. Diallo, who is our network funded investigator. And Dr. Diallo is a senior scientist at the Ottawa Hospital Research Institute, an assistant professor of biochemistry, microbiology and immunology at the University of Ottawa, and also the CEO of Virica Biotech. So with that being said, Elena, I'll stop sharing and I'll turn it over to you for the plain language summary of today's talk.
Thank you very much for the introduction. Good afternoon, everyone. My name is Elena. I'm Senior Research Associate at Ottawa Hospital. And today I would like to tell you about how we use a viral sensitizer technology to increase gene therapy vector production. Genetic therapy is a remarkable and exciting area of biotechnology at the moment. It achieved success in treatments unthinkable 10 years ago. Recent success in genetic medicine paved the path for broader therapies. There are currently more than 800 gene therapies under development. Most of them are in early development, while the rest are in clinical trials. A variety of efforts are underway to apply gene therapy to cancer treatment. Actually, more than 40% of gene therapies are intended for treatment of oncological conditions. While most treatments are still in early exploratory stages, one approach, known as CAR-T cell therapy, had received an approval from US Food and Drug Administration to be used as a therapy in certain groups of patients already. Gene and cell therapy look at illness at the molecular level. The goal is to replace the damaged or dysfunctional molecular components, and gene therapies focus on use of genetic material as a drug to correct the defect, and cell therapies use living cells as a treat for the condition. Delivery of the therapeutic gene could be performed both in vivo and ex vivo when the cells of the patient are extracted and cultured outside the body. For example, in CAR-T cell therapy, the sample of patient's T cell is collected from the blood, and then the cells are re-engineered so they sprout special receptors on their surface called CARs, chimeric antigen receptors. And then these CAR-T cells are re-injected into the patient, and CARs may help T cells identify and attack cancer cells throughout the body. The key step in gene therapy is efficient delivery of therapeutic gene to target cell or tissue. It's carried out by a specific gene delivery vehicles called vectors. A gene transfer tool has to be, first of all, safe. Second, it has to be capable of expressing the desired gene for a sustained period of time, and obviously in a sufficiently large amount of cell population to produce a biological effect. At the moment, the most common types of carrier used in gene therapy are viruses, because they can enter cells and deliver the genetic material. Majority of contemporary gene delivery is achieved by either adenovirus, lentivirus, or adeno-associated virus. Among the delivery vectors for genetically modified cell therapies, lentivirus is the most popular one. There are more than a dozen of completed clinical trials that have used lentiviral vectors for a variety of diseases, including metabolic disorders, cancers, immune disorders, and even rare congenital diseases. The use of lentivirus has been crucial for development of CAR T cells to treat hematological malignancies. Essentially, this treatment became first drug approved by FDA for treatment of pediatric acute lymphoblastic leukemia. There was no therapy available before. It's abundantly clear that the future of viral-based vectors is bright, and the ability to address a variety of human diseases is within reach. But in order to progress and to become available to patients, genetic therapy has to become affordable, has to become sustainable and feasible, especially in a publicly funded healthcare system such as ours. Many challenges still limit genetic therapies from attaining their full potential. The success of approved gene therapies depends crucially on efficient supply of gene therapy vectors. Manufacturing of viruses is very complex and it's very expensive. So not surprisingly, manufacturing is a limiting factor in development of viral medicine. Inefficient vector manufacturing poses a serious barrier for production of genetic medicines. And all technologies that improve manufacturing yield of vectors, such as lentivirus, are crucial for gene therapy. And here, our innovative viral sensitizers technology is a potential solution. Viral sensitizers technology can help overcome the limits and improve the production. Viral sensitizers currently represent a collection of over 100 small molecules. Each of them is capable to attenuate antiviral responses and to improve the production of the virus. Our studies on lentivirus help establish highly efficient production approach. We believe that viral sensitizers technology has a potential to increase the access of patients to new generation of therapies. And with support by BiocanRx, our team developed efficient lentivirus enhancing formulation of viral sensitizers. Our approach is based on supplementing viral sensitizers as a simple media additive upon virus production. Our formulation could improve the production of third-generation lentivirus by 5 to 7-fold on average. Moreover, with support of BiocanRx, the significant progress has been recently made to address the safety aspects of using viral sensitizers for lentivirus production. We had successfully established toxicological stability profiles for viral sensitizers selected into our formulations. And so far, all our findings suggest that using viral sensitizers pose a low regulatory risk for lentivirus manufacturing. So the future indeed will be bright for gene therapies, given the increasing number of clinical studies that are underway, and given the venture capital investments in cell and gene therapy business overall. And yet, regenerative medicine crucially depends on viral systems for gene delivery. On our side, to establish efficient application of viral sensitizers, we currently focus on several more directions. First, on development of efficient analytical methods to be able to track viral sensitizers during the production process and then essentially in the final viral product. Moreover, we are currently extending the application of viral sensitizers to incorporate the production of popular genetic therapy vectors. Development is underway to implement viral sensitizers to production of adeno-associated virus vectors. And at last, but not least, there is an opportunity for viral sensitizer technology to improve the efficacy of genetic therapy, also by increasing the transduction rates of gene therapy vectors. Diallo will talk about all these aspects and finding in a great detail in a few minutes. And I just would like to take a moment to thank all our team members and all our funding agencies. And I would like to thank you for your attention.
Wonderful. Thank you, Elena. So we do have a minute or two for any questions regarding your summary. And if there are no questions coming in through the Q&A, I'll just give it maybe 10 more seconds, then I think we can move on to JS. And so I don't see any coming in. So perhaps JS, I'll turn it to you. And of course, we'll have Q&A at the end of JS's talk. With that being said, oh, and actually there is a question. So I think it'll be addressed to JS, but there will be time for questions at the end of JS's talk, but please do feel free to start asking them during the talk. And that will allow other participants to upvote questions that are particularly relevant to the participants. So with that being said, the one question that did come in is whether or not this is applicable to upstream manufacturing processes, and I turn to you whether you want to address that now or if that's
yeah, I think that'll become clear throughout the presentation. So maybe I'll just jump into that
at that point. Okay, great. Thanks, Jess. Take it away.
All right. Let's see here. Can we confirm that you can see my screen?
If you can make it full screen, please.
It's working on it.
Okay. There we go. Perfect.
There we go. All right. Everybody can hear me fine? Good.
All good.
Okay. All right. Well, thank you, Elena, for that very, very good introduction. I think you've summarized our project very nicely. I'm going to dive a little bit deeper into the science, but I'm going to focus a lot on the viral sensitizers and where they come from. And my hope is that after this presentation, you'll find it interesting on numerous levels because it tells a little bit about the history and also path towards commercialization of technologies, which is a long road. So I'll get started here. Just as a general overview, a few things I'm going to talk about today. Just to start will be a little bit of an explanation around cellular antiviral defenses and innate immunity, leading into the story behind the discovery of viral sensitizers, and then initial observations in the use of viral sensitizers in the context of virus manufacturing. And then I'll talk a little bit more about the analytical and regulatory considerations around the use of viral sensitizers in virus manufacturing. And then I'll take a little bit of time to explain to you our BiocanRx funded project, which really applies to the use of viral sensitizers in the context of manufacturing of gene therapy vectors, and very particular Lentivirus for the use in CAR-T cell therapy, and then touch on the potential benefits for biomanufacturers and cancer patients alike. So this slide here is busy because it's done so on purpose. This is really to illustrate and not necessarily go into very much detail here on the complexity of the cellular innate antiviral defense pathways. So cellular antiviral defense pathways exist really to ward us of pathogens. And they're really our first line of defense when a pathogen enters a cell, including viruses and bacteria, as well as really any foreign genetic material. There are a number of receptors. Some are located outside the cell, some are located inside the cell, but they all essentially serve to capture and detect pieces of these pathogens and essentially propagate a signal using a wide range of proteins inside the cell that will basically trigger a response and the expression of a genetic program. And the genes that are induced when viruses or pathogens enter cells really serve two main functions. They will serve to alert neighboring cells of an infection, alert the adaptive immune system of a pathogen attack, but also they will lead to the upregulation of hundreds of genes that are actually dedicated to actually destroy the invading pathogen within the cell. And so for viruses, that means that the cell is actively trying to destroy the viruses that come inside the cell and try to usurp the cell's machinery to replicate itself. So the reality is that, as you can see from the previous slide, which is comprehensive but still doesn't actually show you everything, the innate antiviral defense mechanisms of cells are actually quite complicated. And really every cell has a certain level of inherent capacity to respond to these pathogens, including viruses, bacteria, and as I said, foreign genetic material. And really, cells express hundreds of genes that are dedicated to this job. And really, they can lead to the destruction of pathogens like viruses. And this is really actually a good thing because these antiviral defense systems are made to be complex in order to defend us against a wide range of unknown invaders. They're really our first line of defense. And unlike adaptive immunity, that line of defense is there to really protect us from anything and everything. And as we know today with COVID and the variants of concern, viruses mutate extremely rapidly. So our cells have to be ready to defend against pretty much anything. And while that's a really good thing for our own health and our own survival, it actually can pose a really significant challenge in the development of virus-based therapies. Sorry. Okay. So what do we mean when we talk about virus-based therapies? Well, acutely today, I think we can appreciate the tremendous impact that vaccines can have in the prevention of infectious disease. And of course, viral vaccines, dating back all the way to Edward Jenner, have had a tremendous impact on global health, and in fact are arguably the most successful therapies that mankind has really ever created. But really, viral vaccines, as established, as mature as they are really the past of virus-based medicines, because in reality now, with genetic engineering technologies, we can now engineer viruses that can grow specifically in tumors, for example, oncolytic viruses, and I'll tell you a little bit more about that soon. And as Elena was alluding to, gene therapies are now promising to cure hereditary diseases that were previously intractable or that would require lifelong treatment. And these are really exciting times for gene therapies. We've seen the approval of a number of gene therapies, including CAR T therapy, as Elena was mentioning, which really involves the genetic modification of T cells using typically a lentivirus or a retrovirus in order to promote killing of cancer cells through chimeric antigen receptors. When we talk about viral medicines and viral therapies, we're really talking about a huge number of researchers and companies that are out there producing, manufacturing, and testing viruses for a variety of different ailments. And from a commercialization point of view, it's very clear that this is a very, very large addressable market. So this brings me to the story behind viral sensitizers, which really takes root in the context of oncolytic viral therapy. For those of you who don't know me, I've actually been in the oncolytic virus field for well over a decade. And oncolytic viruses really have been an attractive technology to me for the simple reason is that they really are exquisitely selective in their ability to grow in tumor cells more effectively than in normal cells. And the reason that they do this is that most oncolytic viruses are actually genetically attenuated in such a way that they can't overcome cellular antiviral defenses. And what that does is it creates a virus that can't really infect normal tissues, but that can actually gain a foothold in tumors. And the reason why that's the case is because cancer cells tend to, reorganize their genomes and actually typically dump a lot of these antiviral defenses that actually help them be detected by the immune system, which tumors don't want because they want to be able to grow. And so there's a natural tendency for tumors to actually get rid of these pathways that would alert the immune system to their presence. And so this is important to realize because part of this means that oncolytic viruses require tumors to actually have defects in their antiviral defense pathways. Unfortunately, that's not typically the case. So even though a large percentage of tumors will have defects in their antiviral pathways, there is a tremendous amount of inter-patient variability from patient to patient. Each tumor is different. Each type of tumor is different. Even within a single tumor, there may be a wide range of different subtypes of cancer cells, even within a tumor. And what that means is that you can encounter, certainly frequently enough, cancer cells and tumors where the antiviral defense pathways are actually quite normal, in which case oncolytic viruses arguably won't work very well because they can't replicate in that context. And just to show some data here to really support that claim, these are results that came from my OHRI lab. And ultimately here, what we did is we took tumors excised from patients and we tried to infect them with an oncolytic virus and quantified the amount of virus that comes out of these tumors. And you can appreciate here on the x-axis in the graph to your left that essentially this is on a logarithmic scale. So each increment is about a 10x change in viral output. And you can appreciate that there's about a 1,000,000-fold difference between a highly susceptible tumor and one that is not. To the right is a similar experiment, but done in a context of a mouse model of cancer, where we've taken a variety of different types of tumors that were implanted in mice, and we've done a similar experiment that we did with the human tissues, human cancer tissues. And again, we see about a 1,000,000-fold difference between your most sensitive and your most resistant tumor types. And that's when we're infecting tumors in a dish, which is really the easiest possible scenario. But you can also appreciate that actually the problem gets much more difficult once you start to try to administer a virus systemically, in particular, where now only in a few instances can you actually detect the growth of the virus. And that's, of course, because there are barriers to delivery above and beyond these innate cellular antiviral defenses that are really what's acting when we're looking in a petri dish. So that's really the reason why we actually started searching for viral sensitizers in the first place. And viral sensitizers is really the result of a decade of multidisciplinary research and development. It initially started with a high throughput screen that I carried out when I was actually a postdoc in Dr. John Bell's lab, now well over a decade ago. And we were really our objective was quite simple. We were looking for small molecules that enhance oncolytic viral therapy in the particular context of resistant cancer cells. Because we were quite aware of this problem, and in clinical trials, it was more than obvious that this was going to be an issue that had to be overcome. So, a little bit humbled by the complexity of the innate antiviral defense pathways, we decided to take an unbiased approach. So we thought to ourselves, tumor heterogeneity is hard to predict. The number of targets that might be relevant may change from tumor to tumor, and we don't even really know which is the most important antiviral target to go after. And so we just decided to take a bit of a phenotypic approach. And essentially what we did is actually what was the first the world first high throughput screen looking for proviral molecules. So that means molecules that actually enhance viral growth, as opposed to antivirals, which was basically what most people have done and continue to do today, for obvious reasons. Now, we went through this process. We screened tens of thousands of molecules, and this is how we identified the small molecules we have termed viral sensitizers. And in those initial screens, we had quite a number of small molecules that really enhance the growth of oncolytic viruses in cancer cells and resistant cancer cells. And you can appreciate that in the pictures on the top right. In control conditions, this is a tumor cell line that's quite refractory to an oncolytic virus here that expresses a red fluorescent protein. And you can see that addition of these viral sensitizer molecules that were identified really vastly improves the growth of the oncolytic virus in this context. So the viral sensitizers were really discovered by high throughput screening, and we were at that time sitting with a number of small molecules, many of which were not really compounds that you would consider putting into people or animals. And so we reached out to Chris Boddy, who's a medicinal chemist at the University of Ottawa. And Chris loves to synthesize new molecules, and he looked at some of the interesting molecules that we had and basically started creating a variety of different variations. And we basically went through several years of collaboration that continues today, where Chris has basically synthesized molecules and we've tested them in our lab and so on and so forth to optimize their activity. And along the way, because we were trying to develop, obviously, pharmaceuticals, we were very interested in toxicology and understanding the analytics and the pharmacokinetics around some of these molecules. And so we recruited the help of Jeff Smith, who's an analytical chemist at Carleton University. And so Chris, Jeff, and I really spent several years doing this back and forth. We had a few beers along the way, which made it fun. And essentially, over the years, we developed over 130 viral sensitizer small molecules through this iteration of medicinal chemistry, structure-activity relationship, and also mechanism of action determination and rational design. And what I can tell you, without going into too much detail, is that these small molecule viral sensitizers really target antiviral defense mechanisms, but in a variety of different ways. We have a variety we have quite a lot of chemical diversity in the molecules that we've identified, and they all have slightly different targets that basically culminate in downregulating the antiviral defenses of cells. So just to state that for the intended effect, these viral sensitizers work quite well. Here you're looking at an animal model where we've implanted a tumor. In this context, the tumor is quite refractory to oncolytic virus infection, which you can see by in vivo imaging. Here we're looking at a virus that expresses luciferase, and so luminescence is observable where the virus is replicating, which here is very specifically in the tumor. And you can clearly see that in the context of the oncolytic virus alone, there's a little bit of replication in the tumor, but really not quite so much. And then when you add the viral sensitizer, you can see very clear tumor-specific enhancement very much in the tumor. And what that means is that if you follow the effectiveness of these therapies, well, as predicted, the oncolytic virus alone in the context of a resistant tumor doesn't work very well, doesn't actually impact tumor progression at all. But once you add the viral sensitizer, now you can see actual therapeutic benefit. So essentially, at that point in time, we had shown that the viral sensitizers could be effective in the context of oncolytic viral therapy, and we're still very much interested in that application. And we were very interested in understanding the mechanism of action. And that's still a pursuit that we carry out today. 130 molecules is a lot to actually handle in terms of figuring out mechanism of action. We've progressed quite a bit, but there's still a lot of work to go. But one of the things that we observed really early on was that the different viral sensitizers belonging to different chemical classes actually could synergize together, likely owing to their different mechanisms of action. And so what you're looking at here in the graph to the right is really a two-way dose response with two different viral sensitizers and at increasing concentration as you go from left to right and bottom to top. And here, what I want you to note is that each individual viral sensitizer actually has a modest impact on its own in this particular case, but the combination of these viral sensitizers really synergizes quite substantially. And here you're looking at a heat map, but really every single point in this graph is actually a viral quantification data point. And what I really want you to appreciate here is that in this red zone, which we call the synergy zone, we're seeing over 100,000-fold improvement in virus output. So this is direct viral quantification that we're talking about. And this particular example, of course, is in the context of an oncolytic virus in a cancer cell, but I think it illustrates the point that these virus sensitizers can be used in combination. And that's something that I want you to retain for the remainder of the talk. So we identified our very first viral sensitizer in about 2008. And in the last pandemic, in at least my living memory, the H1N1 pandemic, I was actually standing in line, waiting for my vaccine, very familiar situation now. And the reason at that time why we were standing in line was not that there wasn't a vaccine, but more that we had shortages because manufacturers produced less than expected by about a factor of 3. And that really gave me the idea, we were sitting with these viral sensitizers, which in combination were giving hundreds of thousands of fold improvement in virus output in the context of cancer cells. So the idea came more or less naturally that maybe we might be able to use these viral sensitizers more than just as oncolytic virus enhancers, but potentially also in virus manufacturing. And so we started that road, as you might expect, with influenza, because it was top of mind at that time. And for those of you who don't know, influenza is typically manufactured in eggs and embryonated eggs. And of course, since we have seen activity of viral sensitizers in animals, it was a natural extension to think that they might work in the context of embryonated eggs as well. And so what we did was actually inject the viral sensitizer at the same time as we were inoculating the eggs with influenza H1N1. And then we basically collected and harvested the virus and quantified by standard plaque assay. And when we did that, we could clearly note a 7-fold increase in actual viral titers. And that might not seem like a lot when you compare to the 100,000-fold that I just talked about in the context of cancer, but you've got to frame that result in a larger perspective. At the time when we were doing these experiments, global capacity for influenza vaccine manufacturing was about 1 billion doses a year. And that's about 1/7 of the entire human population. So a 7-fold improvement in yield means that all of a sudden we have the capacity potentially to actually vaccinate everybody. So practically speaking, these types of improvements in manufacturing output really can make a difference when you start to scale. So those initial observations really led us down a path towards commercialization. We realized that there was a real value add to a lot of different applications in virus manufacturing by developing this technology. And really, it started small, with some initial funding from CIHR, then grant challenges, but then it evolved to larger projects and partnerships with industry. And this ultimately led to the spin out of Virica Biotech in 2018 after an additional funding by BiocanRx, which led us to explore applications in gene therapy, which I'll tell you a little bit more about. And so Virica was spun out of the OHRI really to achieve the full commercial potential of the viral sensitizer technology. As I said, we had good evidence that these viral sensitizers could be used to enhance in vivo viral therapies, particularly oncolytic viruses. But we also have evidence that, as Elena was alluding to earlier, that our small molecules may actually improve the transduction of cells by viral vectors like lentiviruses, AAV, and adenovirus. And in particular, this could be interesting for the manufacturing of CAR-Ts, really at the transduction level when we're producing these cells. But a big driver of Virica's commercial approach has been the application in virus manufacturing, where in some cases we observe over 100-fold improvement in viral output. And I'll tell you a little bit more about that. So as I mentioned, a key driver for this is really the critical need in manufacturing. And this is really across the board. Viral manufacturing is really complex and very expensive. They're not like small molecules. They need to be produced in cells, and that makes it complex. But there are really hundreds and hundreds of ongoing clinical trials, not only in gene therapy, but also in vaccine production. The gap between demand and available manufacturing capacity is really tremendous, and it's only getting worse. Viral medicines are really booming, particularly fueled by the growth of gene therapy. And this is really leaving manufacturers with a bit of a conundrum, because even with planned additional capacity, we won't have enough to meet the needs associated to this growth and interest in viral medicines as a whole. So manufacturing is really a key limiting factor that really needs to be addressed. And that's really where the opportunity for viral sensitizers lie, by actually dampening these innate cellular antiviral defenses in cells. This allows us to actually improve the production of viruses, whether they be for replicating viruses like oncolytic viruses and certain vaccines, or with gene therapy vectors, which are typically used typically produced through transfection of plasmids, which also, of course, trigger antiviral defenses when they are transfected into the cell. And importantly, a differentiating factor here is the fact that this approach is different from what's already out there. I mean, manufacturing has been a problem for a very long time, and the typical approaches that have been used to address that have been improving the growth properties of cell lines with these novel designer cell lines through the optimization of culture media, vector design, and also novel bioreactor configuration to reduce footprint and so on and so forth. And so the viral sensitizers can be an additional tool for manufacturers to improve their yield, and actually can be used in combination with a lot of these other approaches as well, because of the complementary mechanism of action. So we've spent a decade testing viral sensitizers in a range of different applications with a variety of cell substrates, of virus combination. And the thing to retain here is that each cell virus combination is really unique with respect to the interactions that they result in and the antiviral defenses that ensue. And depending on how well matched cells and viruses are, you will get more or less production, you will get more or less antiviral defenses. And this really allows us to customize viral sensitizer cocktails and formulation for each specific application. So in other words, the range of opportunities for testing viral sensitizers and for partnering with biomanufacturers is quite large. So here's an example in the context of modified vaccinia ankara, which is basically in between a gene therapy vector and a vaccine vector. Modified vaccinia ankara is a substrain of vaccinia virus, which has lost a large chunk of its genome and is quite defective in its ability to overcome antiviral defenses. And MVA is used as a platform. Well, first of all, it's a smallpox vaccine. It's approved for emergency use as such, but has also been explored quite extensively as a cancer vaccine platform, as well as a gene therapy vector. And what we did is actually tested viral sensitizers and identified a viral sensitizer that could enhance the production of MVA in a wide range of substrains. So MVA exists in a variety of different substrains, and they don't all behave the same. And when we tested our viral sensitizer in the context of MVA, we saw a range of impacts, all the way from as low as 5-fold improvement in yield up to 20-fold improvement in yield. But on average, we were seeing about a 10x improvement in yield. And so what we did was take one of these average strains that gave about a 10x improvement in yield at the small scale, and we actually tested the impact of the viral sensitizer in a scale-up production system called the iCELLis. And for those of you who don't know the iCELLis, it's a very neat process intensification technology currently sold by Paul, which actually allows you to grow adherent cells on macrocarrier strips, which basically are nanofibers that permit the growth of cells really in 3 dimensions. And in a nutshell, that allows you to take an adherent cell culture space of meters square and pack it into a tiny bioreactor the size of a toaster oven. So we actually used the viral sensitizer technology in the context, and compared to control conditions, what we observed is that we could actually boost production in that context by about 4-fold. And really, I think this example is important because it illustrates a few features of viral sensitizers in this context. The first is, of course, you're going to have a variety of different impacts depending on the virus strain that you're going to get. That you're working with, and of course that really depends on the interaction of the virus and the producer cell. So you do expect some variability there. The other point to make is that, of course, this approach is scalable because we started at a very small scale, in this case 96 well, and moved all the way to a 4-meter square bioreactor, and we could still see the improvements at that scale. The final thing that it illustrates is really the compatibility of the viral sensitizers with these process intensification technologies like the iCELLis. And so answering the question that was posed earlier, essentially these viral sensitizers are really just added upstream. So whether you're using a suspension bioreactor or an adherent cell culture, the idea is that you add the viral sensitizers during the manufacturing of your virus, really in the upstream process. And actually through studies that Chris, Jeff, and I performed a little while ago, we now know that the viral sensitizers are actually metabolized quite efficiently over a period of 24 hours during viral production, such that there's actually very little left by the time you harvest your virus and you start your purification process. And again, through efforts by Jeff Smith in particular, we've developed sensitive assays to actually track and detect these small molecules and even some of their metabolites in the manufacturing process. And what we can say is that there is no detectable viral sensitizer at the end of a standard purification process. And to illustrate that, I'd like to show this figure, where essentially we took that same viral sensitizer enhanced MVA process and tried to detect the viral sensitizer at the end following a standard sucrose cushioning purification. And so in panel A here, what you're seeing is actually our capacity to detect the viral sensitizer using multiple reaction monitoring, which is a mass spectrometry-based approach that's extremely sensitive. And at least in our hands, we could detect down to femtomole level quantities of viral sensitizer if it was present. So in A, what we did is we actually took an MVA purified sample and spiked in about 1 microgram of the viral sensitizer, and we could actually detect very easily through that nice single peak the presence of the viral sensitizer. Now, in the context of B, where we actually use the viral sensitizer upstream to boost production, and then we followed a standard purification procedure and we tried to detect the viral sensitizer at that point, we could not detect it. And again, I'd like to point out that our assay actually has femtomole level limits of detection. And that's really important because viral sensitizers as a strategy for manufacturing enhancement would be considered as a product contact raw material. And what that means is that this is a product that's in contact with your virus during the manufacturing process, but that's not actually meant to stay with your final product. It's purified out. And the regulatory requirements for this type of product are quite clear. And in fact, you may be aware of certain things that you might already be using in your processes. Things like suspension cell bioreactor anti-foaming agents, for example, fall under that category. And even certain salt buffers also actually fall in that category. And what's required for those types of materials, these product contact raw materials, is really to demonstrate that the viral sensitizers are below a certain threshold based on toxicological assessment. And such very sensitive analytical assays of this type are really critical for that. The other requirement, of course, is that the viral sensitizers be produced in GMP quality. So that leads us to really the BiocanRx Enabling project, which is again a continuation of a very fruitful and long-lasting collaboration between Dr. Bodhi Smith and my lab, and now in collaboration with Virica, as well as the Ottawa Hospital Biomanufacturing Center, and very specifically the virus manufacturing facility. And in this project, essentially, we will be developing and scaling up a viral sensitizer formulation really catered for the production and enhanced production of lentivirus, again, with CAR-T in mind. And my group, Dr. Bodhi and Dr. Smith, will be focused on our lentivirus viral sensitizer process optimization and really formulation development and optimization. And Dr. Smith in particular, and as well as Dr. Bodhi, will be focused on analytical assay development. And as I showed you before, we have already very good methods using multiple reaction monitoring. The only issue with that approach is that most biomanufacturers don't really have access to this level of mass spectrometry. And so we're trying to develop assays that can be more easily transferable to manufacturers, things like simple HPLC or gas chromatography. In this project, Virica is contributing heavily through the provision of viral sensitizer molecules and the development of toxicological and regulatory packages, as well as scale up and GMP manufacturing of the viral sensitizers, which, as I mentioned before, will be required for clinical manufacturing of viral vectors. And finally, the virus manufacturing facility with the infrastructure that it has will help us scale up a lentivirus process using the viral sensitizer, and also provide test materials so that we can track and provide, again, supporting evidence that the viral sensitizers are effectively eliminated using their purification procedure. So just a hint of how we've done this. Essentially, I've showed you this before, and I gave you a little bit of a flavor of what that looks like when we combine viral sensitizers together to actually improve viral output. And we basically took a very similar approach here, but in the case of lentivirus, and here we're using, as Elena mentioned before, a 3rd generation lentivirus, which is produced in this case in the context of adherent HEC293 T cells through triple transfection using PEI-based transfection protocol. And essentially what we do here is actually use and leverage our high throughput screening capabilities, which we've really perfected and developed over the past decade, to really rapidly test a lot of different viral sensitizers alone and in combination. And essentially what we found is that a combination of viral sensitizers actually leads to kind of a synergy zone where at the small scale, we're seeing upwards of 7x improvement in lentivirus output. And we've recently translated that to a larger scale, in this case T75, but we've now gone up to T175 and seen a very similar improvement in yield. And now the hope is that we can actually scale this up to fit the biomanufacturing process at the virus manufacturing facility at the OHRI and provide materials for Chris and Jeff to develop their analytical methods. So I just want to finish on a few points here just to illustrate the potential benefit of this technology. I would highly recommend that you go see another webinar by David Bohonek from Millipore, where they did some very interesting modeling using a software called Biosolve. And for those of you who don't know what this is, Biosolve is really just a massive Excel spreadsheet that automatically calculates the impact of process changes on virus production costs. Is a really neat software we've started playing around with as well, that essentially allows you to figure out the impact of process changes on your cost of goods. And David's analysis has been very, very interesting because it really does suggest that in certain contexts, very modest improvements in upstream manufacturing yield can actually lead to very significant changes in your cost of goods. But ultimately, really what it means is increased production capacity. A 5x improvement in yield can have a really tremendous impact on accessibility of viral vectors for a variety of different applications. And of course, CAR T is what we have in mind here. I just want to impart that CAR Ts are really expensive to manufacture, and they're significant. The manufacturing costs are really significant proportion of the actual costs that the healthcare system pays for each dose of CAR T. And the lentivirus component of that manufacturing actually is about 20% of the total manufacturing cost of CAR T. And so a 5-fold improvement in yield could really have an impact not only on the production capacity, but also on the cost of goods, which ultimately means that, and we hope that, more patients would benefit from CAR T, which really has taken the cancer therapeutics field by storm, and we're all excited about. And I know that BiocanRx really believes it. So with that, I'd like to thank you for your attention and of course, entertain any questions.
So JS, maybe while we're waiting on questions to come in, I'd just like to thank you and Elena for a really, really great presentation. I think that really, excuse me, told the story very well. It shows what it takes to bring a product like this to commercialization. And so thank you for that. We do have a few questions coming in. So the first question actually is more of a comment. It is very clear presentation. Thank you. Can you speak a bit more about how VSEs affect purity of viral yields compared to virus manufacturing without VSEs?
I'm trying to unmute, sorry. Anyway, it's a very good question. Thank you for that. So that's a complicated question because it's really quite context dependent. So the way you have to think about this is, of course, when you're producing a virus, a lot of the contaminants that you're going to get in your end viral products are going to come from the cells. Cellular DNA and things like that. These are things that manufacturers really often struggle with. And in reality, the higher the yield of virus, the lower the concentration of the contaminants relative to that virus can be. And so, even though we're hoping to test that actually with this project, we don't have direct evidence of that, but at least in theory, this is what we would expect. And the other thing, obviously, is the viral sensitizers, because that's the added component. And as I've shown you before, very simple purification procedures will really eliminate these very small molecules quite well. And that's because they're orders of magnitude smaller than the viruses that we're trying to produce. So it's really easy to separate out.
Great, thanks, JS. And a few more questions coming in. So next question, beautiful talk, very exciting work. In the context of OVs, would these sensitizers also enable infection of normal cells in vivo? If so, how can this be mitigated by careful dosing?
Yeah, that's a very good question, one that I've had many times. So, this is an important point. Really, a big part of that is the choice of the viral sensitizer that we use and how we deliver it. So currently, our approach is to use these viral sensitizers through intratumoral administration, which is really the largest, the most frequent mode of administration of oncolytic viruses that are out there. That's the lowest hanging fruit. That said, we're very interested in developing approaches to actually selectively target viral sensitizers towards tumors. And I can go on about that, but maybe I'll save that for another conversation. Now, that said, really, we do, when we treat with viral sensitizers, it's a very transient thing. It's not long lasting. And that's actually the nice thing about it, because the alternative in oncolytic viral therapy is to actually genetically modify the virus to be more effective, which once it's in the body, you can't really do anything about that. But at least the viral sensitizers, they have that transient nature, and they'll be effective really for a period of 24 hours, giving the virus enough time to do its job. But they're not meant for continuous administration.
Perfect. More questions coming in. So I think we're good on time, actually. So this is fantastic. So next question. Thank you for, no, sorry, that one no, I did not. Thank you for two very interesting talks. I am interested in the manufacture of VSEs themselves. Is this done by a third-party GMP manufacturer, or is this part of Virica's work?
Yeah, so Virica is certainly coordinating those activities, but yes, the molecules are being manufactured by a very reputable CMO that has GMP capacity.
Perfect. And next question. You spoke about using multiple VSEs for a synergistic result, and also about the need for GMP manufacture of these VSEs. Do they differ greatly in how they are manufactured? Can they be manufactured simultaneously?
That's a good question. So the answer is that they're obviously different molecules. So their manufacture, their specific chemical synthesis is going to be different. Now, that said, most of the molecules that we're working with are quite simple molecules. Often they're one-step chemical syntheses, and so they're really easy to manufacture. Can they be done in parallel? Absolutely. But this is really just a requirement of whether the CMO can manufacture multiple batches, and we've selected a CMO that certainly has that capacity. Perfect.
Great questions, everyone. Thank you for adding them, and thanks, JS, for answering all of these questions. So next question, do the specific VSEs and combination of VSEs work similarly in the production of different viruses, or are particular VSE combinations better for certain viruses than others? For example, lentivirus specific viruses versus AAV specific VSEs.
Yeah, that's a good question. As I said before, it does come down to the pairing of the virus and the cell line. And so, for example, when we're developing a formulation for a lentivirus, the expectation, again, is going to be very specific to the context. Here, we're talking about in the case of the BiocanRx project, an adherent 293T third generation lentivirus PEI-based transfection. What we'll optimize to work for that should work for other similar lentiviruses of a third generation produced in a similar way, because ultimately it's the system as a whole that you have to look at. Now, will the same compounds work in the context of HEC293 in the production of AAV? Not necessarily. They might be a different set. There may be some overlapping molecules that work in both contexts. And ultimately, a big part of what we do is leverage our high throughput screening capability to really just go and nail down what is the best combination for that specific application. And, on the Virica side, that's certainly been what our partners are really interested in doing, because everybody's manufacturing process is different. It's amazing how many conversations we've had and how varied the processes are, even for the same virus. And so that customization and that capacity to customize is really kind of a key thing.
Perfect. And I see I missed one of Tommy's questions, so sorry about that. The question was, excellent work. Knowing the mechanism of action for viral sensitizers, could you generate cell lines that recapitulate the same boosting effects? Is Virica also interested in the generation of new cell production models?
Yeah, that's a very good question. So I'm a little bit biased here. Obviously, we're developing a small molecule technology. And the reason why we're doing that, and we think that's preferable, is because cell line generation is really quite labor intensive. There are designer cell lines out there, but frankly, they've been around for decades, and there really have not been many approved products actually manufactured using those designer cell lines. It's really hard to make a designer cell line. The other point is our viral sensitizers, because they're so broadly, and I think they work so well because they're so broadly acting, you would really need to knock down a lot of different genes to actually elicit the same type of effect. And I should mention that it's also possible that some of those genes may actually negatively impact cell health, in which case your cell growth will be compromised. And so that's why we feel that the transient viral sensitizer approach is better. So, of course, we're interested in production platforms that have improved productivity, but that is not an area that we're moving into currently at this time.
Too many options out there for you to go down.
You've got to pick your battles, and lots of other groups are really attempting to make manufacturing cell lines that are genetically modified. But as I said, seeing the challenges that were faced by companies like Crucell and Vivalis, it's a good business model, but it is challenging because it needs to be very broadly applicable, right? And that's that can be a challenge.
Great, Jess. So a couple more questions are coming in. The next question is, do the specific VSEs and combinations work similarly in the production of I'm sorry, I just asked this one. From a commercialization perspective, is it clear you seek therapeutic product partners? Can you talk a little bit more about what an ideal partner might look like? Is it safe to assume you might want to grow with a partner in very early development, or can you offer a strategy for supporting comparability, for instance, to adapt an existing manufacturing process to use your VSEs?
Okay, I think there's multiple questions in that question. So I think in terms of the therapeutics approach, this is one that's obviously near and dear to our hearts. But fundamentally, we can be, the viral sensitizers can be an adjuvant to a variety of different therapeutics. And ultimately, we're looking for a partner that would really have a need to develop that platform for enhancing their therapeutic approach, because I think in that context, we would be really co-developing a therapeutic and bringing combined value, right? Because some therapies, as I've shown before, some oncolytic viruses may not work in a large, variety of patients because they can't overcome those antiviral defenses. And so we think it's a real value add to bring in these viral sensitizers. And so we'd be really seeking co-development partnership, and so partnerships where both parties can bring in some resources. On the manufacturing side, well, we now are working with a lot of different partners, and we have different ways of doing that. One way is that we often send the viral sensitizers, or a selected subset of viral sensitizers, out to clients. But that's only really feasible if clients actually have the capacity to do this more high throughput screening and the large scale evaluation. Oftentimes we actually leverage our high throughput screening capabilities to develop formulations for clients. So we do a little bit of both. Megan, I think you're muted.
Has to happen at least once, right? So that does look like it concludes the questions. No others are coming in, and we do have a few more minutes. So if it's all right with you, JS, I'd love to ask a question maybe for the HQP who are on the call. And if you could just give maybe a couple of your lessons learned as you develop this product, whether it be at the early stage, I know, where do I start?
Where do I start indeed?
Yeah, maybe a couple of points just for our HQP audience, or our grad students and undergrad students.
Okay, well, so I'll tell you one thing. I mean, the lesson learned for me is perseverance. Perseverance is the key here, and don't get discouraged by data that's not good, or don't be discouraged by an approach that you thought was going to work and then all of a sudden doesn't, or isn't being accepted by your peers for whatever reason. I mean, I just think back of those early days with Dr. Bell, when we were so excited about the viral sensitizer application for oncolytic viral therapy. And at that time, oncolytic viruses were not yet an approved therapy. So this was still a pipe dream. And, when we were trying to propose to develop this for oncolytic viral therapy, literally people were laughing us out of the room. And, we stuck with it. We stuck with it. And, in 2015, the first oncolytic virus was approved, and now this is now front of mind. And of course, the manufacturing aspect, that you always got to be on the lookout for what need your technology or what you're doing can actually be helpful for. Sometimes you lose sight of that, right? You're very focused on your project, and you're not really thinking of the big picture. So I certainly would encourage you to think of the big picture. And also don't be afraid of commercialization and working with industry. There's a tremendous wealth of knowledge and expertise out there. And really, there's no better way to actually get your research to have an impact. We need more people doing that. We need more Canadians to do that, actually. And I'm glad to see BiocanRx really be front and center in promoting that.
Appreciate it, JS, and I appreciate you taking that question. I think it's one that comes up a lot in our HTTP community, so I appreciate you having that point of view. And no question, your data certainly supports what you just described, so that's great. Maybe one last question that did come in as you were answering that. So for boosting activity of AAVs in vivo, would VSEs be injected with the AAV, or are the VSEs used only in the AAV production process?
No, so this is an area that we're very excited about. Actually, there's a lot of interest in using the viral sensitizers in combination with AAVs, and my development plans on that front really focus on the development of a co-formulation. So the idea would be that we would have a viral sensitizer really co-formulated with the AAV, such that there's a lot of AAV gene therapy applications where you're administering the AAV either in the eye or in the ear, or in very localized regions. And I think there's a real opportunity there to avoid systemic delivery. I mean, systemic delivery, as was alluded to earlier, comes with a lot of problems, right? You're basically treating the entire patient with the molecule. You don't necessarily always need that or want that. And so in that context, I think as a starting point, at the very least, we would favor these kind of more localized co-formulation type developments.
Perfect. Okay, and I'll just give it another 30 seconds. Again, thanks everyone for the questions. I think it made it a really interactive session, and I think it just begged to the point that JS, you did a wonderful presentation. Elena, I think you set it up very well. So we did get a question coming in the chat box that if you're okay staying for a few extra minutes. Just having some technical issues, so it's difficult to see the questions coming in, but it is, simple question. Have you tried cloning some VSEs into vectors?
Well, we can't do that because they're small molecules, so we can't clone them in. That said, we've certainly been thinking about the targets and using things analogous to VSEs to clone into viruses. But you can't actually clone a viral sensitizer because they're small molecules.
Okay, great. So I think with that, thanks again for answering so many questions. I think that closes out this session. And again, just one really big thank you to both of you for presenting in this series. It was really excellent. So thanks again.
No problem.
And thanks, of course, to all of our participants. Always, JS, you guys are always welcome. Thanks, everyone. Take care. And thanks to the participants as well. Great questions.
Thank you for your interest.
Bye, everyone.